participate in our cycle of seminars. So thank you very much for
Thank you.
with us. Thank you, Iván. And as always, I will say just a few words. In this case, I will go a little bit faster. So let me tell you that Iván graduated in the Universidad Autónoma de Madrid in condensed matter physics. In 2001, he got his PhD degree in the same university, and he was co-supervised by Marisela Vélez and also by Professor Philippe Debeau. And his research was focused on lipid asymmetry, the flip-flop of ceramides, as well as the biological implications of the enzymatic conversion of its sphingomyelin into ceramide. I think that we can claim that your research has always been focused on model membranes and proteins.
Yeah, exactly.
Living in membranes and so on. So this is the topic you should take home. In 2006, he joined the research group of Paco Monroy at the Universidad Complutense, and he also makes some research on the mechanics of model lipid membranes under the action of different proteins involved in biological processes such as apoptosis or bacterial cell division. In 2013, he was awarded with a starting grant, and in this case, he focused his interest into the fabrication of biomimetic systems for in-cell ATP synthesis and delivery. After this grant, ERC grant, left him the opportunity to develop his career as PI, first as a Ramón y Cajal fellow and then as an associate professor at the Universidad Complutense. From 2014, he leads the mitochondrial membranes laboratory, also at the Universidad Complutense and Hospital 12 de Octubre. The projects within this lab are essentially devoted to the reconstitution of fundamental mitochondrial proteins in model membranes to understand the dynamics and self-assembly under out-of-equilibrium conditions. Well, important contribution has been made with the rotatory motor, protein Atp Synthase, as recognized by the Spanish Biophysics Society with the prize of the Spanish Biophysics Society SBE 14. Well, finally, currently, and in partnership with Frédéric Pincet, Vivek Malhotra, and James Rothman, Iván has been awarded with a ERC Synergy Grant to decipher the formation and self-organization of the protein secretory pathway. So please go ahead. Thank you very much.
Okay, thank you. Thank you, Jose. Well, I would like to thank you, Jose Miguel, and the organizer of these seminars to invite me and let me the opportunity to present some of my results done in my lab. And the topic I am going to talk about today is related to the self-assembly of ATP synthesis in lipid membranes. And before we get into this topic, I would like to present also my team and the main focus on the main interest of our lab, which is trying to understand how lipid or membrane, I would say membrane, and protein interactions can contribute to the shape of different organelles. Because you have, in this picture, you can see an electron micrograph where you can distinguish two different organelles where the shape is very funny, which is the Golgi here, represented as, formed by different stacks that are mainly composed with different proteins and lipids and following a gradient across one direction, which is the cis to the trans Golgi. And also on the side, you have also mitochondrion, where you can also distinguish the double membrane and some structural hallmarks of the organelle, which is the formation of these cristae that are responsible for allocate different proteins responsible for the synthesis of ATP in our body. So before we get into the topic, I would like to also to present some different projects we are running now in our lab, and I also wanted to present the people involved in those experiments, just to present them because they are doing a great job now. So we are interested in how Golgi genes, which are the proteins surrounding the Golgi stacks, how they are self-assembled into model membranes, like here, and we are following that with different techniques like AFM. We are also working on the proteins involved in the process of fusion in mitochondria, because mitochondria here, we will see that later, is not a static organelle, but they fuse and divide constantly along the cell cycle. And you can see that we can also study fusion process in model membranes. As shown here, you can see how two membranes become one of them by the action of these proteins. And the main topic of this talk and the main research we have been running all these years, or this last year, I would say these 5-6 years, is the action of this protein, the ATP synthase, that I will present in a couple of minutes, and how this action of the protein can have a contribution to the remodeling of membrane shapes. And also this slide allows me to tell you about the techniques we are using. So we have already seen AFM. We are also working with fluorescence microscopy, and also now we have been incorporating molecular dynamic simulations to trying to understand a little bit more about the mechanisms involved in our results we are seeing with the experimental results. And finally, I would like to say that with this small presentation that we are a group working in model membranes, as Jose Miguel reminded. So what people call now in the synthetic biology field at the bottom-up approach, meaning that beginning with a very complex membrane, what we are trying to do is to take the different single components and reconstitute it in model membranes, and with a minimal set of components trying to study the function and the structure of self-assembly of these proteins in those model membranes. And this is not new. I mean, this is comes from the 60s or 70s, where they called reconstitution. So we are working with this kind of problems. So the main question is how these proteins can remodel membrane structure. And we are involving those proteins present in mitochondria and Golgi. The tools, have you seen that they are mainly based in fluorescence microscopy, and the system is mainly based also in model membranes as a form of giant vesicles, as you can see here, or supported by layers or different membrane models. And to understand what I'm trying to convince along the presentation, I would like to present some mechanical parameters that are important to understand how the proteins can shape the membranes. And for this, you have to consider that a membrane is a two-dimensional fluid that can be deformed, suffer some deformations, and each mode of deformation implies an energy scale that is governed by a mechanical parameter. So you can think about a membrane like a fluid that can be bent in this way, can be straight, meaning that the membrane can be changed, the area can be compressed or can be straight, or even can be sheared, meaning that this kind of deformation is how if the membrane is solid or fluid. And in those modes, the main contribution of the shape would be represented by this bending mode, meaning that how difficult it is to bend a membrane. And you can also have in mind that the membrane are like a piece of paper. So it's very easy to bend. It's very more or less difficult to stretch. You can take a sheet of paper, and it's very difficult to stretch the paper. And it's very easy to break, to shear the membrane. So this analogy, I think, is quite good to represent how a membrane behaves from a mechanical point of view. And the parameter that gives you the energy scale is called the mechanical parameters, like the bending modulus, here represented with the letter kappa, which is around 10 to 20 kBTs. So this is the order of energy scale of the bending. And the total energy cost is given by the how do you separate from the equilibrium, right? Represented here by the spontaneous curvature of the membrane. So if the strain is bigger, the energy involved is bigger, and if the strain is smaller, so the other way around. But the energy scale is always a scale up with the bending rigidity. The same is for the stretching energy. So you can also be far away from the equilibrium area, and then you can stretch or compress. And this gives you also the strain of the deformation, but it's also governed by the mechanical parameter, which is the stretching modulus. And in this case, this is the capital K here, and in this case, the number is the typical number for a membrane is 100 millinewtons per meter. Okay, and these numbers, these mechanical parameters, as a material parameter, is dependent on the composition of the membrane. So at equilibrium, this is mainly governed by the composition of the membrane, the composition or the state of the membrane, of the state of the main components of the membranes. I say that because you can think about a fluid membrane made of, for example, a typical fluid lipid like POPC or DOPC, or below the transition temperature, above the transition temperature, which is in a fluid state. So in this case, it's very easy to bend and, let's say, easy to stretch. But if you go from this fluid state, to a more condensed state, just by dropping the temperature, you end up with a very more gelled state, and it's difficult to bend. It's more difficult to bend, and it's more difficult to stretch. Okay, so then you can play. You can play with all these strains, mechanical parameters, just to reproduce or to induce shape change in the membrane. Okay, so here this this is my first paper, indeed, where we could promote a shape change from a prolet shape of the vesicle. This is a gel vesicle made of PC mainly, and just by incorporating some molecules out in the outer membrane, so changing this spontaneous curvature, we could induce this phase shape change to a budded shape. And then when these molecules go into the inner layer, so the difference, the curvature spontaneous goes back to a more natural value, then there is a shape change which is reversible. Okay. So the question we are in this topic, I'm going to focus in mitochondria and the action of the protein I will present in a few minutes, the Atp synthase. And for this, I just want to remind that this picture, which is very frequent in the textbooks, is not quite real. Because yes, we have a single mitochondria here represented by a typical size of several angstroms in this direction and this direction. And we can also recognize, as I told you before, the double membrane, and the inner membrane is also very many invaginated to place a lot of proteins doing their job in this case, so to produce the ATP of cells. But if we go to a more real picture, a dynamic picture, these are cells where the mitochondria are labeled with a specific dye, fluorescent dye, and you can see that these mitochondria are not single units, but forming a network, and this network is very dynamic. So when you see this, you ask yourself that there is something that should be happen to deformate in that way, to be very plastic like this, no? So, and how this plasticity can be affected, or can, not affected, but can contribute to the bioenergetic function of the organelle. And in particular, so we are zooming a little bit up to the protein of the study. And those cristae, those invaginated membranes, there is where the electron chain is located. And this electron chain is composed by different complexes, from complex 1 to complex 5. And you all know that these proteins are mainly, what they are doing is to create a gradient here of a protein gradient across the inner membrane, following different electron transfer between the different complexes. And this proton gradient is dissipated by the last one, which is the ATP synthase, the complex 5. And this electron gradient, proton gradient is used for the protein to create the ATP in our cells. So you know that this ATP is the, let's say, the biochemical molecule, energy molecule for proteins to do work. In particular, this protein, it's a molecular nanorotor. So this is why in the title there is this word. It's a molecular nanorotor composed of different subunits, and this protein is, have different properties. The first one is that in the membrane, you have two different parts of the protein. One is embedded in the membrane, which is the rotor, and also there is a very soluble, big domain of the protein that catalyzes the reaction from ADP to form ATP. And the way it's formed is working is because of this gradient, proton gradient, or electrochemical gradient also, that makes the rotor to rotate. And because this part, the soluble one and the membrane one, is linked through a stack that is not centered along this axis, but is shifted a little bit, this shift promotes this change in the conformation of this part, and to open and close these pockets, catalytic pockets, to produce the ATP. But the funny thing of this protein is that it's working in this direction to form ATP, but it's also working in the other direction, hydrolyzing ATP. So you can fool the protein with ATP, and then you promote the rotation of the protein in the other one around, okay, in the reverse mode. So this is the question we address is, just looking at this picture, is if this movement of the protein is able to produce something in the membrane to remodel the membrane. Okay, and also something that it is known up so far is the location of this protein, so the ATP synthase, in the cristae. So this protein in mammalian cells, so this is a top view of the protein. So here you will see a ring, the rotor of one protein, and here is the rotor of another protein in the top view. And you can see also that there are many other subunits around this rotor that can interact each other. And this biochemical interaction is responsible for the dimerization of this protein in the cristae of mammalian cells. So finally, this interaction is responsible for this dimerization that promotes the protein to be at the edge of the cristae, at the rim of the cristae. So the question we address is, okay, this is governed by the biochemical interactions, and what happens if there is a physical interaction between proteins mediated by the membrane that can also produce these very, very long structures. And because and due to the rotational movement of the protein. So the main question we are addressing in the last years was if rotation of the protein can modulate or can mediate this interaction to gather the proteins, and the gathering of the proteins produce these highly curved regions and accumulation of the proteins at the curved regions. That was our main question during the last years. And to do that, and this is, I starting now telling you about experimental details, experimental approach and details and results. To do that, we go back to a different ATP synthase, which comes from bacteria, from E. coli. We're able to extract from membranes and to put into middle membranes. But the main important thing of this protein from these organisms is that mainly the domains responsible for the dimerization in mammalians is not present in this protein. So that means that it's a very useful model for us because we can delete the biochemical interactions, and if there is some action of the if there is some effect produced by the rotation of the protein, we can allocate this effect just only because of the rotation, but by the presence of particular interactions. So the protein here lacks these domains that are responsible in mammalian cells for the dimerization at the tips, at the rim of the cristae. Okay, so this is why we choose this one. And to do experiments, we have to put the protein in a functional state in our model membranes. So our membrane is based on GRVs. So this is vesicles which are micro-sized vesicles. So they are big enough to be visible under microscopy. And the first thing we have to prove is that we are able to put the protein in inactive conditions. So this is the model, and you can see that there are many ingredients in this model, and I will try to explain to all of you all of this to you. So this is the green orange is representing the GUV. So this is a lipid bilayer of about 10 to 20 microns. This is represented in orange because we label the UV with a fluorescence probe, a fluorescent lipid probe, and then the proteins are incorporated from the outside. And because this is very soluble and this is very hydrophobic, we think that the main orientation of the protein is as represented as in the picture. So because you have to put it's more it's easier for the protein to penetrate the membrane if they face the transmembrane domain before. And then the vesicles are fabricated in a way that it contains another fluorescent probe, which is called the pyranine. And this is a probe which is sensitive to the pH, meaning that at very low pHs, the fluorescence is lower, and at basic conditions, the fluorescence is higher. Okay, so just because it becomes protonated, and the fluorescence properties are changes because of this protonation. And then to trigger the activity of the protein, what we do is to fabricate the vesicles with a low amount of cations here, represented by potassium cations here inside, and we load the vesicles, the outside of the vesicles, with a very high amount of potassium. And then by addition of this active transporter, which is valinamycin, it feels the gradient of potassium from here to here, and it's transported across the membrane, the potassium, to compensate the charge of the potassium gradient across the membrane. Doing that, we can create an electrochemical gradient across the membrane, which is responsible to activate the protein. And doing this, we can start trigger the action of the protein, which in the outside there is the substrate of the protein, ADP and phosphate. They transform into ATP, and this reaction is coupled by the transport of the protons, and this basidification of the inside of the vesicle is seen because of the presence of the preanin increases. I don't know if it's a little bit, I mean, there are many ingredients in this model, but this reaction is coupled to the proton translocation, and so the direct way to see the formation of ATP is just to see the increase in the pH increase in said vesicles. Okay, and this is what we do, and this is what we got. So here there are two different, just as a representative cases of different vesicles, but here the first one contains the protein here, and the second one doesn't contain the proteins. So what we did is to add this valumanizing, and then you can see that there is an increase in fluorescence inside vesicles, whereas there is not in vesicles that do not contain proteins in the membrane. So this is our proof that we can put the proteins at work in this kind of models. And the second thing was to see if the action of the protein, the activity of the protein, meaning the rotation of the protein, could favor the self-assembly of those proteins into curved regions. I remind you that the main question is this one. So is the rotation of the protein able to induce a curvature sensing, a curvature seeking of the proteins into more curved regions? And the state-of-the-art experiment now is this pulling lipid nanotubes experiments, where you let the protein to choose. To choose where the protein prefers to be. So you present the protein as a lipid tube, which is representing a very curved region. You can see that the cross-section of this lipid tube would be a very curved region, a very curved membrane. And then, at the same time, the protein can be also in a UV, which is 10 micron scale, and that means that the curvature is very low. In those experiments, what we did is to label the protein with a fluorescent light probe, label the membrane with a fluorescence lipid probe also, and then do a ratio of intensity. So you can see here that the experimental parameter is how the protein is, or the lipid, in this case is represented about the protein, how the protein is concentrated in flat regions represented by the UV or in curved regions represented by the tube. So to do that, we microaspirate a UV, and then with a second pipette and a bead surrounded by streptavidins, and the lipid composition contain biotin lipids, biotin functionalized lipids. So we approach the bead into close to the vesicles, and then when you separate the bead, there is a tube that is formed, and in that moment, the protein can choose between to prefer to be in one side of the vesicles or on one side of the filament. And just by taking the ratio of the intensities in each channel and in each location, meaning the surface, the UV or the filament, you can get a number. In this particular representation, sorting S parameter equals 1 means that the protein doesn't prefer anything. So it's the protein is happy to be in the flat region or in the curved region. And written in this way, sorting parameters bigger than 1, meaning that the protein prefers to go to the filament. Okay. So what we do is to measure the intensity along the tube, as here. So we can see here different conditions, and also across the membrane, okay, of the vesicle. And here are the results. So here we present the sorting parameter as a function of different conditions. The first one is to control just lipid to lipid. So the membrane doesn't contain any protein. So we label the membrane with two different dyes, one red, one green, and you can see that if the membrane composition is made of typical lipids where there's no intrinsic high spontaneous curvature of the lipids, the sorting parameters is 1, as expected. We also did some experiments, control experiments with another protein, which is bacteriolabsine. I will tell you a little bit more after. And you can see that the sorting parameters is 1. So that means also that the bacteriolabsine doesn't prefer any curvature region. I mean, doesn't prefer any of those regions. And then here is the main results where we can follow if the protein wants to stay at the curvature region or at the flat region. So here in cyan is the ATP synthase, and you can see that there is not that high curvature sensing, and when there is no ATP in the medium. So when you put some ATP here, what we are trying to do is to activate the proteins, and when the protein is active, you can see that the sorting parameter increases a lot. I mean, that is 2.2 on average. And this is a direct measurement to prove that the protein prefers to be there only when the protein is rotating, okay? And you delete this rotation by specific inhibitors, like DCCD, even in the presence of ATP, you go back to the previous state that the sorted parameter is still 1, okay? So this is a direct proof that the action of the protein, the movement of the protein can lead the protein to be in a region which is more curved than in the flat surface. This curvature depends on different, so the question that raised now is why the action of the protein is promoting this curvature sensing or curvature sorting of the protein to the filament. Why is this? Why the action of the rotation, which is in this plane, can affect the self-assembly of the protein in different regions. And to answer the question is we have to go to this model that explains the curvature sorting parameter as a function of different parameters involved in the system. The first one is the curvature of the filament. So we can, so represented here as C, we can pull different filaments with different radii. So you can expose the protein with different curvature. And also it depends on the surface area of the protein, here represented A, and the curvature, the intrinsic curvature of the protein. This is telling you how the shape is, how is the shape of the protein. So if you have cylindrical shapes of protein, it doesn't want, by geometry, it doesn't want to be in a curved region. But if you have this kind of asymmetric shapes of proteins, edge proteins, you can think about the possibility of the protein to be to prefer to be in a curved region. So this is stated here in the spontaneous curvature of the protein, and then you have something that is related to the membrane, which is the mechanical parameter of the bending energy of the membrane, as I told you in the first slides, which is represented by kappa, this kappa. So kappa lipid, kappa I represents in the absence of proteins, so the bending rigidity of in the absence of protein, or when the bending rigidity changes because of there is proteins. Okay, so you can imagine that these two function. This is an exponential function with a positive exponent, and this is an exponential function with a, let's say, positive or negative. In principle, there is a negative exponent here. So this expression gives you an idea of how this sorting parameter depends on the shape of the protein, and then when it is enriched, the filament is enriched with the protein, how this enrichment or the content of the protein can modify the membrane, the bending properties of the membrane. So here, the first exponential just is a coupling of the shape of the membrane with the protein with the curvature. Okay, so you can think that there is a preferential curvature if the shape of the protein is fits very well or match very well with the curvature of the membrane. And this is why I represented this picture of the protein, where you can see that the sum of the control made with the bacterial rhodopsin protein is a very cylindrical protein. The curvature of the protein is almost 0, and there is no preference for any curvature. It's a very, let's say, cylindrical protein, so it doesn't want to be in any curved region. Here, for the ATP synthase, as presented here, you can see that there is some. You can calculate the spontaneous curvature of the protein, and this is not very high, so it's 0.02 nanometers to -1. So that means that the protein is neither a very sharp shape to be allocated to a curved region. So this is not a contribution that is important in our experiments because of the intrinsic shape of the protein. We are working with proteins that are not very asymmetric. In contrast, the second part of the exponential, the second exponential, you can see that there is a ratio here of bending modulus. If, for example, kappa is bigger, this kappa in the presence of protein is bigger than the kappa in the absence of proteins, this is positive and this is negative. So that means that when you enrich membranes with proteins, the fact that there are proteins in the membrane, the membrane becomes stiffer. And if it is stiffer, this is negative, so meaning that when you populated your membrane with stiffer inclusions in the membrane, it is a penalty energy cost to be sorted in curved regions. And this is what happened to other proteins, where you can see that at the beginning there is a positive sorting, because the protein fits into a particular curvature which matches with the shape of the protein, and then when it becomes very enriched, the concentration of the protein is very, very high in that filament. It's reducing the sorting because the membrane is very stiff. In our case, this contribution is almost 0 because, as I told you, the shape of the protein is very cylindrical, so the contribution to the sorting is not very important. But because we are obtaining an experimental data which is positive, which is 2, we thought about the possibility that the protein could induce, the action of the protein induce a softening in the membrane. And I remind you that a softening here means that in the presence of proteins, kappa is lower, and this exponential becomes positive. So contributing to the sorting of the protein to the filament. And this is what we observed before with other techniques, but we can characterize that also with the microinterpolation experiments, where you can pull a giant vesicles into a micropipette and then aspirate and see what happens to the geometrical shape of the vesicle. From the geometrical, simple geometrical parameters here as the radius of the vesicles and the distance of the tongue of the vesicle inside the pipette, and the difference of pressures that aspirate the vesicles, you can obtain directly the mechanical bending of the system. And you can see here that you can represent the tension because of the aspiration as a function of the deformation, just by seeing that how the membrane is aspirated, how much is aspirated. You can plot these typical strain deformation plots, and from the fit of this data point, you can obtain the bending rigidity. So here the results with lipids with ATP synthase and ATP synthase under the action of ATP and the presence of ATP, so in rotating conditions. And you can see that we obtain typical values for the membrane, for a membrane, which is 10 to 20 kBt, as I told you in my very first slide. But this is parameter is reduced by a factor, let's say 3, because of the action of the rotation of the protein. So under non-equilibrium conditions, under the action of the protein, we can see that the protein became softer. And this is producing a membrane softening that is responsible at the end of the protein sorting in the regions, in the curved region, in the filaments. And this is just to visualize that in a more visual, way. And you can see, so on the top you have a protein, a vesicle with the protein reconstituted in the membrane, and when the protein is active. So this is why I put that on. So I put the voluminizing, and the protein is active. In the bottom panel, you can see this. I mean, not the same, but the same reconstitution, the same experiment, but when the protein is off. The only thing I do is to inject the buffer, but without the volumizing that triggers the activity of the protein, as you can remember. And you can see very well that beginning with a similar state of fluctuations, when you activate the protein, the fluctuation of the membrane are very high here, as compared by the when the protein is off, is not active. So this is the visual way to visualize, the direct way to visualize how the protein produce this membrane softening, because the membrane can fluctuate more because it's softened. Okay. And to ask and to answer another question is, okay, we can put active proteins in UVs. We can measure the protein sorting into curved regions because the protein is active, the protein is rotating. We can say why is producing this protein sorting, because the membrane is softer. But how this softening comes? Remember that this kappa, this mechanical parameter, is a material property, meaning that it's mainly composed, the mainly contribution to this value is given by the the membrane composition. Here we are not changing anything. We are not changing, I mean, they are the same protein content and the same lipid composition. So why this kappa is changing? And to go into this, to trying to understand what's happening at the molecular level, we did some molecular dynamic simulations where we can really see what happened to individual molecules. And what we do in collaboration with the group of Manuel Melo in Lisbon, we could put some in a small piece of membrane. We put 4 ATP synthase. We delete for practical reasons, we delete the soluble, the F1 domain, the soluble domain, and we put that at work, meaning that we force the protein to rotate, applying a torque, and then we run the experiments, the simulations, in the absence of this torque, let's say at equilibrium conditions, and under the action of the torque, or under the action of the rotation of the protein. And you can see here the difference that are producing when the proteins are rotating, in this case. So you can see that the fluctuations, very easily to see that the fluctuations of the membrane are increased in the case of the rotating case, under rotating conditions. And that reproduced very well what happened in those experiments. So exactly the same. Can put that again just to see. And you can see not only that the fluctuations of the membrane are increased, so producing this membrane softening, but you can also see that there are some, I would say, interacting contacts between proteins, because it's not a dimerization per se, but there are sometimes that the proteins wants to be together. Here, for example, or here, as compared to the control, where the proteins are always kept at the same system, more or less at the same distance between them. So fluctuations also produce the protein to gather together. Okay, and with these movies, we can extract this information, and we can say what happened to different properties. So this is just to represent two things. So here, the first one is how long the protein stayed together, and you can see that there is an increase under rotating conditions. And also here is no, this is the other way around, right? So finally, I don't really remember very well, but at the end, what we see is that rotation promotes this kind of kiss and go interactions. So proteins become more frequent to be together, but it's only in a less period of time, and then they go apart, and then they can to be together again. And this is what this represents, these two panels. But the most important thing is that we could also extract the structure changes in the membranes to support the membrane softening during rotation. Here, what it is represented is the area per acyl change of lipids around, across the distance of the protein. So we took every protein, every single protein, and then we measure the acyl change area at the distance of the protein. So here in this axis is 2 nanometers apart from the protein, 4 nanometers away from the protein, etc. And you can see that in those, in both leaflets, in the upper leaflet and in the bottom leaflet, you can see that there is an increase in this area per acyl change at the vicinity of the protein. So meaning that around the protein there is an expansion of the lipid packing on the membrane. And at some point, when the lipid is very far from the protein, you can recover the same lipid packing for all of the lipids. So you can see that in the rotating conditions, in orange, there's an expansion of the protein. So finally, we get a whole picture where we can conclude that we can put ATP synthesis in UVs under conditions. The active mode of the protein promotes the monomeric proteins to be in curvature regions just by the action of the rotation of the protein. And then to explain why this rotation can promote this protein sorting in filaments, it is demonstrated that the rotation produces a membrane softening, and that fits with the theoretical models that are reported in the literature. And the softening is supported by a membrane expansion around the protein, at the vicinity of the protein. So it's like this movement is perturbing somehow the molecular packing of lipids around the protein. And we think that these observations can, well, we are trying to convince the biological community to, as a side effect of the rotation, because don't forget that the main function of the protein is to produce ATP in cells. But we think that this movement, this rotation, can participate at least to the self-assembly of these proteins to the rim of cristae. And we used this model, like in the specific interaction, biochemical interactions, to really allocate the observations to this effect. And finally, I would like to thank, of course, the funding agencies that support our research. And yeah, here there are many people in different projects we are running now. But those that are participated more directly in this, what I have been presented so far is Bilayer, which is in this institute, Manuel for the molecular simulations, and in Paris, by the very first part, well, we were able to put the protein. It was the first reconstitution of the ATP synthase in UVs. And I would like to finish just by claiming that, saying that we are looking for a postdoc. We are offering a postdoc position to do microscopy, time resolver fluorescence microscopy with proteins involved in Golgi, so related to the Golgi project. We have problems to get people involved, so you know somebody that wants to participate in those projects, please tell him, tell her, and to contact me, please. And that's it.
Okay, thank you very much, Iván, for your excellent results and talk. So I think it's time for questions, and the last question chat of this cycle. So I encourage you to participate, to make questions. So Maria.
Your nice presentation, and I was just wondering what was the lipid composition of the groups you were using, because Atp Synthase is perfectly linked to the presence of cardiolipin within the mammalians' mitochondrials.
Yeah.
And I was just wondering whether the composition, the complex composition of the lipid bilayer may have to say something on the events you have observed.
Yeah, there are some, for the same reason we choose the ATP synthase from E. coli, we also choose a lipid composition that is very neutral. We only working with, for the functionality assays, we took a lipid extract from E. coli containing cardiolipin, just to improve the activity of the protein. But for the last part of the presentation, for the protein sorting, we remove all these lipids, and we were working with POPC only, just to separate, to discard any other effect that could be contributing to the protein sorting, because it's been already shown that cardiolipin also prefers to go to cover regions. So just to discard these effects, and to fully demonstrate that the rotation is also maybe participating in this protein sorting, we didn't work with cardiolipin. In the?
Head groups or acid chains?
Yeah, of course. Cardiolipin prefers to be in curved membranes. That's have already shown. But that would hamper, with our results, because we didn't know, we could end up with some results and saying, well, it's cardiolipin that's driving the protein sorting to the filament, or is, so just to really allocate the observations with the particular rotation, we remove all these contributions.
Okay. Yeah. Thank you.
Yeah, more questions?
Well, thanks very much for the talk. It's really nice.
Thank you.
And I have two questions. It's more curiosity and probably coming from ignorance. So in your first slides, in one of them you show this network between the mitochondria, which is something I have never seen before. And is it known what is this network formed with?
Yeah.
So how?
So this network is made of, in fact, mitochondria are filaments. It depends on this is very dependent on the cell, on the type of cell, okay? But in this case, for example, in fibroblasts, these are fibroblasts, these filaments are made of, mitochondria are filaments that can fuse and can divide a long time. And this is produced mainly by the fusion machinery of the mitochondria, which is composed by different DNA-like proteins. For the outer, you can see that you have to fuse and divide a double membrane. So you have to orchestrate that, to fuse the first layer and then to fuse the second membrane. And for this, there's a machinery for doing that, which is composed of, for example, in mammalian cells, is mitofuscins for the outer membrane and OPA1 in the inner membrane. And there is the other protein to produce fission, which is DRP1, I think. So yeah, it is known. And this is made to, it's very, so this dynamics is very important to control the bioenergetic requirements of the cell. And also it's also participating in, if there is a mitochondria that doesn't work anymore, there is excluded from the network and goes to the mitophagy. And yeah, it's impressive. It's a very complex
pretty
system.
Cool.
Yeah.
And my other question or curiosity is regarding, you show on one of your first slides as well that you add protein to a membrane system and then you remove it. And I wonder, how do you remove the protein from the membrane once it's inserted?
Here. Here, it's not a, yeah, it's not a protein. It's a molecule. It's a ceramide. So here,
okay.
so ceramide is a lipid, a very phobic lipid, and they insert first in the first monolayer, and then by flip-flop, they go to, they equilibrate to the second, to the inner leaflet, and yeah, but it's not a protein. It was a, yeah.
Thank you.
Thank you very much for the excellent talk, Iván. A question, maybe it makes no sense, but I'm going to ask it anyway. So I was wondering, because all this occurs in the inner layer of the mitochondria, and I was wondering if this like movement can affect to the mitochondria itself, like being more like the plasticity or the elasticity of the mitochondria.
Yeah, we claim also in our papers that this, for example, this membrane softening that we have measured independently using two different techniques. Here we can obtain from this fluctuation, also we can obtain the bending modulus of the membrane. And we can also, we have also obtained a membrane softening because of the action of the protein. So independently here and those experiments. We were very happy because with two independent measurements we obtained the same results. So that means that the kappa is reduced by a factor of 3. And we claim in all of our papers that if you have a softer material, you can straight or you can deform this material easily. So if you want to produce these events where the filaments are very dynamic, or to produce these invaginations, these cristae, it's better to have a material that is soft than stiff. So yeah, how, I don't know, because finally the whole system is very complex, but at least the membrane is softened when the action of the proteins are there. I'm very convinced that not only the ATP synthase, but all the movement of the proteins can also do this kind of things. So in particular to the inner membrane, is very enriched in protein. The protein to lipid ratio is very high, I think. So, yeah.
Thank you.
Okay, more questions. Yeah, I have a comment. When I see the ATPase as a whole, it is very easy for me to consider this as a nano machine with an element, a rotating element.
Yeah.
However, when considered as in more molecular aspects, when I try to visualize the interaction between the amino acid side chains embedded in the lipid, in the hydrophobic core of the lipid bilayer, when this element is rotating, which kind of interactions permit such dynamical behavior while embedded in the membrane?
So finally, my view is that the membrane is a fluid also.
Yeah, in fact, I
Yeah.
consider the hydrophobic part of the bilayer as a lubricant for.
Yeah, exactly. This is what I want to
However,
mention.
this is not.
Yeah, and I mean there are two scales. No, at the molecular level, you can see, and this has been also been reported that cardiolipin binds very. The lifetime of cardiolipins around ATP synthase, at least in mammalian, is higher than other lipids. So there is a specific interaction of cardiolipin to the. So and this is. And it has been proposed that cardiolipin could be also a lubricant for the rotation, but this haven't never been proved yet, because it's very difficult to prove the lubricant effects in such a small scale. But then it's a fluid, so as a fluid it can be, the fluid can transmit different interactions along the membrane as a fluid. And we are working on that also because we are trying to see what happens to the diffusion of proteins when the protein is active or not. And we have some nice results that the fluid can serve as a propagating media for the proteins to self-assembly into different states.
As far as I know, and correct me if I'm wrong, the phase behavior of cardiolipin is highly regulated by calcium.
Yeah, I think so. Yeah, in mitochondria.
Yeah, in mitochondria. Yeah. Well, no, essentially even in model membranes also. So, did you somehow analyze or study calcium as an element in?
No, we haven't tried that. Never.
Yeah.
No.
And a last comment. I don't know if it is something useful for your studies, but it is currently possible to prepare GUPs composed of two membranes?
Yeah, that's a dream. As far as I know, nobody has been able to do a double layer in a controlled way.
Yeah.
Because you can do UVs and you can with different techniques, and you can in the heterogeneity of the sample you can find vesicles inside vesicles, but in a controlled way this is not yet possible.
It could be useful for your.
Yeah, of course, because you can mimic the mitochondria. We are trying to do something in that direction, but very at the very starting point now.
Okay.
But yeah, we would like to do that.
Okay,
Yeah.
thank you. So there are no more questions. Thank you much again for your
Thank you.
for this answer to our questions, and let me give you a small present for your participation in the Roca Solano, and just to thank you again. You are here with us. Thank you.
Thank you very much.
Bueno, pues fenomenal, excelente. Muchísimas gracias.
Nada, vosotros.
Carolina, veniros a comer.
Pues...
Díselo a Pili.
No, Pili no está.
Ah, que Pili
Pili no
veía.
está, sí,
Oye,
sí.
no había gente, está aquí.
Perdón, no sé qué he dicho entonces.
¿No se nos escriben todavía o ya no?
Ya
No, ya no nos oyen, ya está todo cerrado.
No lo podía oír al principio.
Al principio. Bueno, espero que se haya solucionado.